Thank you. Yeah, so as Lindsay says, I'm Ali, I'm the R&D manager, and I'll just jump straight in. So I'll go over a bit about the background of Atelerix, who we are, and how our technology works, the science behind on to the products that we offer, and how these can help you with your organoid cell models, your and your tissue to develop your sort of in vitro models and such. And for each one of them products, I'll go into some of the data from either our own internal development or from partners or customers that have provided us with data just to give you an idea on what we can do with our technology. So at Atelerix, we offer an innovative approach to the storage and transportation of fresh biological materials, so cells, tissues, viruses. And we do this, we approach these cells as the fragile, sensitive, and living entities that they are. We do this by encapsulating in an alginate-based hydrogel. They protect the biological products from biochemical and physical damage, whilst retaining the viability and function without the need of harsh preservation methods such as cryopreservation. So, as I mentioned, this is a alginate-based hydrogel. So this is derived from seaweed, and our products are of pharmaceutical grade. They have been highly characterized and are consistent. We've developed these products and optimized their formulations and formats for different biological samples. So we have products that go from as small as a 2 ml tube up to a large bag format. And we also, in between that, we have larger tubes. We have in plate preservation, so from 24 well plates to 4 well plates. We've also streamlined these protocols so they're easy to use and scalable, as well as being quick for both parts of the process. So the encapsulation, this goes about 10 to 20 minutes. And then on the other side, when you want to get your material from our alginate-based hydrogel, you have a simple cell-friendly salt-based buffer that you add to the gel, and then this turns it into a liquid and you can retrieve your material. So briefly to go over the sort of the science behind it, our technology relies on hypothermic temperatures. At hypothermic temperatures, the cells go into the metabolism and the metabolic activity, enzymatic activity decreases, essentially putting these cells into a state of hibernation or sleep, which enables the storage and shipment of these biological materials in sealed vessels. Ordinarily, without our technology, if you were to reduce the temperature from physiological temperatures to hypothermic temperatures of these biological materials, it would lead to the damage and degradation of the lipid membrane. Normally, lipid membrane is a fluid kinetic structure, but when you reduce these temperatures, they go into a static state, leading to protein deconfiguration and pore formation, which ultimately leads to damage and then cell death. With Atelerix technology and encapsulation, we essentially protect these structures, which allows us to extend the shelf life of these fresh biological products. After this storage time or shipment, and you want to retrieve the biological material, as mentioned, you add this cell-friendly salt-based buffer, which turns the gel into a liquid, and then you can retrieve your material, return them to normal culture conditions, where after a recovery period, they'll be in the same state as what they were when you entered storage. Listed here are some of the cell types that we have successfully preserved using Atelerix's technology. This is not a complete list, so if there is something that you are interested in that you don't see on this list, then get in touch and we'll see what we can do to help. So going into sort of how we can assist the generation of in vitro models and such. the generation of advanced cell and tissue models is becoming increasingly important with the phasing out of animal models, especially for such things as drug discovery. So there's a greater demand for the sourcing of materials to generate these models, such as human tissue samples, as well as a greater demand of these advanced models themselves, whether they be organoids or engineered tissue. So here I've just mapped out a very simplified version of the process of getting from, say, a patient to the generation of these models, and labeled the different products of that Atelerix have that can assist in the preservation of materials at each point. And I'll go through the rest of this presentation, highlighting each one and where we can help out. The first of these is for the preservation of tissue samples. So tissue that has just been, say, taken from a patient, and you want to preserve the tissue for later use, or you want to ship that tissue for other people to collaborate and generate models. But first, what are the problems that we have with tissue preservation? Well, you're removing said tissue from its normal physiological environment. As once you've removed the tissue from this environment, the condition and the viability of this tissue decreases, which means that you need the model generation or the experiments need to be carried out as close as possible to the retrieval. This unfortunately isn't always possible, as the sites of retrieval, say hospitals, are not always near the site of the end user or the labs. With current distribution methods to enable transportation of these samples from site to site are not really up to standard. This picture on the left here is some tissue that we received that we used during the beginning of our development for Tissue Ready. And as you see, it's just tissue in a tube with some basic medium. So improvements in tissue preservation methods would extend the time tissue could be used post-retrieval to get higher quality results and more reliable data that can be used for, say, model generation, cancer model generation, patient-derived organoid development, and advanced screening. So this is where Tissue Ready comes in. Tissue Ready can assist in the storage and shipment of primary tissues, organoids, spheroids, and microtissue. So I'll just briefly go through how the process works. When you get Tissue Ready, you'll get 3 tubes. There'll be a tube with gel-based beads and a tube with gel A. You add gel A, the gel-based beads. This causes the gelation process to start. During this time, add the tissue to the tube, and then the gelation will complete around the tissue, encapsulating it, and this is where you get your preservation in this state. You can then store or ship this tube as you see fit, and then once you are ready to release, you use that 3rd tube, which contains the dissolution buffer, inject that into the gel, which will turn it into liquid, and then you can retrieve your tissue easily from this liquid state. We actually have 2 products in the Tissue Ready line. We have Tissue Ready, which we provide a concentrated version of gel A. So with this concentrated version, you would dilute it with a medium of choice. So say you know your tissue prefers a certain type of media or has certain additives that you would require. You simply add that media to the gel A, and then you continue the process as previously described. We've also developed or recently released Tissue Ready Plus. With Tissue Ready Plus, we have developed a media to add to gel A ourselves for optimal conditions, so in optimal results. So if you don't have a preference on media, or you don't know what is required, then we would recommend going with Tissue Ready Plus to get optimal results. So what are the advantages of Tissue Ready Plus? Well, obviously we preserve tissue at hypothermic temperatures, and this enables the shipment of these valuable and fragile tissues, extending the shelf life, which would increase the global reach of these tissues. Looking at the other end of it, on the other side, the cells that you may want to take from these tissues to use for your generation of your models, the quality and yield of these cells would be is greatly increased with using tissue ready as, say, just leaving them in medium and shipping them as I showed in that picture previously. The final point is to enhance the flexibility of workflows. So we can't always control when we receive these tissues. I've worked in labs in the past where we are waiting on surgeries to finish, so then we can go get the tissue, and these can happen at 5 o'clock at night, and then you having to work into the night to get your work done. With tissue ready, you can give yourself that flexibility and maybe do it on a Monday morning instead, which is a bit more preferable. I'm sure everybody would agree. So just to go into some of the data that we've got with Tissue Ready. So this is actually with Tissue Ready Plus. This was done in collaboration with a clinical pathology lab in Newcastle, Novapath, who kindly helped us out with this. The process is just listed along the bottom here. So essentially what we did was the tumor was resected from the patient. It was then given to the clinical pathology lab, Novapath, who went on to preserve this tissue in Tissue Ready. So they stored the tissue for 2 days at room temperature, and then this using the dissolution buffer, and then went on to process the tissue as would be normally as would be processed the standard, so fixed, paraffin embedded, and sectioned. These sections were then stained and then assessed by a blinded clinical pathologist. So he provided a score to essentially give it an integrity score, which would then how well it would be used for, say, diagnosis. So here's just one of the sides, one of the tissues that we use. We use multiple tissues in this thing, but I'll just present one of the ones that we got, which was for cancer liver tissue. I'll go through this now because this is kind of follows a similar trend throughout all of the data that I'll be showing, where we have non-stored. Now, non-stored is our fresh tissue, our fresh cells. These are your DAISY or your control, essentially. And we also have medium controls, which are the same process as Tissue Ready Plus, but just media. So they go for the same temperature at the same time, but there's none of our hydrogel technology. It's just media on it, so like in the tube that I showed earlier. So if we look at the top with the H&E and compare the non-stored with Tissue Ready Plus, you can see that the histological integrity is retained between these two sections. Whereas if you look at the medium control, you can see that the slide isn't as good, and you're starting to see some artifacts. This can be shown in this graph on the right, which is the histological integrity score. So this is the score that was given by the blinded pathologist, where 10 is the perfect score. Between 5 and 10 is good enough quality. You could use it still. Anything below 5, you start to get into the questionable sort of state. And what you can see is, non-surprisingly, non-stored, where it's just freshly come out of the patient, is 10 across the board. And then after 2 days at room temperature and tissue ready, you can see that we are largely, well, we are from 5 and above consistently, with an average coming at around 7.5. Whereas if you just have media on its own, you can see that you're getting some drop into that lower region when you're saying just on that borderline of 5. Additionally, you can get you can see that the markers, the immunohistochemical markers, 81, 83 here, that's a cytokeratin cocktail, which is used as a marker for carcinomas, and Chi67, which is a proliferative marker. You can see that in tissue ready, these markers are retained as they are with the non-stored tissue. As I said, this is just one of the tissue samples that we've done. We have released a few months ago a white paper kind of highlighting the work that we did with NovaPath, where all of the other data or all the other tissue types are shown. So if you've got an interest in this, then go onto our website and you will be able to find that white paper. And this one here is with one of our customers who've kindly provided some slides for us. What they wanted to do was to preserve mouse liver samples, tissue, and for 7 days at refrigerated temperatures. And what you can see is that from the non-stored 3 days and 7 days, the liver morphology remains intact and structurally sound. So those two examples kind of looked more at the structure of how the tissue is preserved. If we want to look at more of a functional aspect of preserving these tissues, we can look at isolating cells from tissue. So in this case, from preserved cancer tissue. This is again done with the clinical pathology lab at NovaPath, and follows the same process pretty much as what we did last time, with getting the sample from the patient, preserving in tissue ready. But in this case, we had the sample shipped to an oncology research facility at the University of Sheffield, where it was, after 4 to 5 days, it was released on their side, and they took the sample and then isolated some cells, expanded the cells, and fixed them for cytokeratin 7 as a marker of cancer cells. So these were done using esophageal cancer tissue. So the graph on the left is showing viability of the cells that we are getting out. So this is kind of showing the quality of the cells from the tissue. Non-stored, so the fresh tissue, we're seeing sitting at about 80% viability. If you compare this to tissue ready plus, well, they're getting very comparable results. Whereas if you looked at the medium control, the blue bar, you can see that the viability drops significantly. So the quality of the cells that we're getting out isn't great. If you're looking at the actual yield of the cells, the graph on the right, you can see that the number of cells that we're getting out is comparable between the tissue ready plus tissue and the non-stored tissue. This is looking at both healthy cells and cancer cells. So the bar at the top is the solid bar at the top are the healthy cells, and the stripy bar underneath are the cancer cells. And you can see again, both healthy and cancer cell proportions are about the same. So we're not just protecting the cancer cells or the healthy cells, we're actually protecting both populations. Whereas again, looking at the blue bar, looking at the medium control, if you just leave them in media, then you can see that the yield is significantly reduced for both populations. And this can be visualized on the images on the right here, where you can see the sort of blue purple cells are the non-stained healthy cells, and the brown are the cancer cells, the CK7 positive cells. And you see the comparisons between tissue ready plus and the non-stored versus the negative or unhealthier cells on the medium control. So we move along this diagram, we go into, and you've got your cells, your associated cells. I'm not going to go too much into this one, but we have a product as well to preserve these cells at room temperature, ambient temperatures, hypothermic temperatures. And we have that product called CytoStore. So you can ship these cells using our hydrogel as well. And the process for doing this is very similar to that of tissue ready. You get given 3 tubes. one of the gel-based beads and one of the gel A. You mix your cells with gel A, add the gel A with your cells to the beads, and then the gelation process and the encapsulation process will occur. And again, at the other side, you add your dissolution buffer, inject that in, and then turn the gel into a liquid, and you can retrieve your cells. Here is some of the data that we have with some cells with being stored in cytostore. So we have them at 7 days in cytostore at various temperatures depending on the cell type. So we have MSCs on the left, HeLa cells in the middle, and human airway epithelials on the right. So we have 100% cell yield, obviously, for the non-stored, and this is, anyway, we're getting around that 100% mark. We're getting a similar cell, viable cell yield with all of our cell types here when stored for 7 days in cytostore. Whereas if you look at, again, the blue bars with the medium control, you can see that you get a significant loss of cells when just preserved with just standard medium. The final part, the actual models themselves. So we've kind of diverted this into two separate areas. The top one being if you've generated your organoids from your cells and you just want to ship these organoids in a tube, you can do so using the first product I described, Tissue Ready, which works in much the same way as how I described it. You simply put your organoids in your gel A, add that to your beads, and then you're ready to go. Your organoids in a tube, ready to ship. But if you want to create more of an assay-ready model, we have multi-well. We have a format for multi-well plates. So for organoids, engineered tissue, or even if you just have cell monolayers, you can use well ready to encapsulate these models and ship them at hypothermic temperatures. So why would you want to do this? Well, there are a few problems with preserving advanced models. Well, as we are getting these models towards more physiological temperatures, physiological relevance, they are becoming increasing in complexity. And with this increase in complexity, there's an increase in the fragility, both in terms of viability of these, sensitivity and viability of these cells, as well as their actual function. And this has led to some of these models being pretty much incompatible with cryopreservation techniques, which means that these models are unable to be shipped to end users. So you're not being able to get them sales, say if that's what you're doing. And if you were to get these models to sites, say drug discovery or drug discovery sites, there is a high level of skill and technical awareness needed to generate these models. To say you ship out iPSC cells and they need to cultured up to their cell of interest. These are long culture times, sometimes up to a month, require a high level of skill and knowledge to get to that, to get to them cells of interest. And even if you get them, to get them consistently and repeatable and good quality is sometimes a challenge. So this could lead to limited uptake of these physiologically relevant models. And this is where well ready comes in. So this is suitable, like I say, for cell monolayers, organoids, and microtissues. And again, this is a hydrogel-based system. So you get your gels. We have 3 gels. Essentially layer these gels and add a gelation buffer to cause the gelation of the gels. Remove any residual gelation buffer and give it a quick wash to get rid of any last remaining bits. And then it's essentially ready to ship. These 3 gels have different functions. So we have gel A, which is what encapsulates the biological material at the bottom. So this is our cell protective layer. This is what provides our biological protection. We have gel B, which is slightly more viscous, which is our shock absorbing layer. This is what provides our physical protection from transits. It's thrown around in a box on its way to its destination. And we have gel C, which is essentially a seal layer, which just seals off the well. So what advantages do we have of well ready? Well, this enables the shipment of complex, physiologically relevant and fragile cell models in an assay-ready format. So you ship them out, they get to their end destination, say again, it's a drug discovery unit. All they need to do is release them from the gel and then throw on their drug of interest, and then they're ready to go and we'll get consistent results. Which leads on to the second one of getting high yield, so high viability and good quality cells that will produce these repeatable results. You also have this, again, like with tissue ready, a flexibility of workflows. So you can, if there's an unanticipated problem, you can pause the experiment and come back to it in a day or two without losing the cells that, like say, can sometimes take up to a month to generate. Increasing the shelf life of these of this biological material again can increase the global reach, letting you reach markets that you were previously unattainable. And finally, reduce the requirement for these expensive complex shipping methods, such as use of dry ice or liquid nitrogen. So I've got two models to go into with well ready, one with organoids and one with some engineered tissue. I'll just go through the organoids first. So what we did was we got commercially available mouse iPSC derived hepatic organoids and cultured them as to the supplier recommendations, which in this case was in Matrigel. When we had got them, when they were matured, we encapsulated them using well ready and stored them at room temperature for the hepatic organoids. This was for 5 days. After the 5 days, we released the organoids from the wells, returned them to culture for recovery, and then did any endpoint analysis. So we looked at viability and function of these organoids. We then compared the data to the non-stored controls. So again, these are the ones, the fresh ones. These are before the encapsulation, the day zeros. So the first thing we wanted to look at was viability. So on the left, we have ATP-mediated luminescence. So this is what we used as an indicator for the viability of the cells. So the first bar, the purple bar, is the non-stored, day zero, no culture, just straight into our viability assay. The next one on day zero, the purple one, is these cells return to their standard culture conditions of 37 degrees. We then performed it again just to see that increase. You can see the increase as it's still likely proliferating. The next two bars are after 3 and 5 days of storage in well ready, and then a further 3 days of culture under standard culture conditions. And what we can see is the viability and activity of these cells is comparable to the non-stored cell organoids. The images on the right show the structure and morphology of the organoids being retained after preservation and well ready after 5 days compared to the non-stored organoids. So, and as well as the viability of the cells, we give more visual representation of this, where the middle one showing the live cells on the right, you can see that there are no clusters of dead cells in these organoids. Obviously, structure is one thing, but if we're going to be using these models for their function, we want that function to be retained as well. So in the case of these liver organoids, we looked at their drug metabolizing capability. In this case, we looked at cytochrome P450-182. So again, on the left, we have day zero with 0 hours of culture. And then the next bar being day zero, and then 72 hours in physiological conditions, 37 degrees. And then the next two bars, 3 and 5 days of storage, and then 72 hours of recovery culture. And we can see from that the P450-182 activity is very similar or comparable between the non-stored and the 5 days of storage with 3 days of culture. We also want to look at the marker expression. So we selected CK19 based on supplier information, and we can see that the markers are still retained after 5 days of encapsulation in well ready compared to the non-stored. The final bit of data that we have actually comes from one of our customers. So we can, so these top ones are looking at sort of cardiac models. So these are engineered heart tissues composed of iPSC-derived cardiomyocytes and human ventricular fibroblasts. And they stored them for 2 days at room temperature using well ready. And the top 2 graphs there show that they recover their contractility behavior, whereas the bottom one shows that the tissue responds to drugs in a similar fashion to that of non-stored tissue. The bottom graph is with muscle. So these are engineered microtissues from primary human skeletal muscle, and these were stored for 3 days at room temperature. And much like the liver organoids, you can see that they required 3 days in culture culture for recovery to get back to their full contractility behavior. And that's all of the data and hopefully all the process gone through. So in summary, Atelerix can support every stage of the process from sourcing fresh tissue to getting fully developed organoids or cell models to customers, to collaborators across the world. So we do have some extra resources available. So we, as I mentioned previously, we have a tissue ready plus white paper that looks at some of the cancer tissues that I haven't presented here. It also looks at the RNA, the dual DNA in situ hybridization models and RNA isolation. There is also an organoid white paper which goes more in depth into that them liver organoids that I've mentioned. And we've also recently released a data pack which includes all of these tissues, cell models, viruses that are listed here. So if you're interested in any of that, or interested to see what else has been done, then go find that data packets on our website, and yeah, hopefully there's something there that takes your interest. So I just want to say thank you for joining the webinar, and thank you for listening. And I will now pass over to Lindsay for questions, which I'll be happy to answer.